Hello, everyone. We are here today with Adriana Romero, who is a postdoc at the University of New Hampshire. And today, Adriana is going to tell us about her latest publication entitled Soil Metatranscriptomes under Long-Term Experimental Warming and Drying. Fungi allocate resources to cell metabolic maintenance rather than decay. That you published recently in Frontier in Microbiology. So, hi, Adriana. Nice to have you here.
Hi, thanks for having me. I'm excited to talk about this paper.
So, yeah, what can you tell us about this paper? How all this started?
Okay, so this paper started during my PhD. This was actually one of the side projects that I was working on during my PhD. At the end, it wasn't included as a chapter, but it was something that I was working on since 2015. And this paper started because Kathleen Treseater, my PhD advisor, whom is the lead author of the publication, her and some of her colleagues had this long-term warming experiment in Alaska, basically some greenhouses in the middle of the forest that they were passively warming the soil. And in my area, which is, I guess, fungal soil, we're very interested in understanding how fungi are adapting to climate change. Mm-hmm. Well, we want to know first, are they adapting? So that's the question. Exactly. And then we wanted to know, okay, so how are they adapting? But to actually answer that question, we first need to know how are they responding to begin with to environmental changes. And that's how the idea started. So Kathleen told me, we have these greenhouses in Alaska. They've been warmed for like 10 years. What do you want to do with them? And I told her I want to see how fungi are responding. And at that time, there had been studies, DNA-based studies. So basically, how is the community shifting? Who's there? How is the community changing? But I saw that no one had worked with the metabolism of the fungi and how the metabolism is changing. So that's how I got interested in this. I wanted to know how the metabolism of the fungal community was changing under warming.
So basically, you extract the whole RNA of the soil and then sequence it. But then how did you select it, the genes of interest that you, for example, you have been working a lot with the genes that code for carbon degradation enzymes.
Yeah.
Like this. So how was your criteria to select the genes? Because I suppose under your treatments, you have a lot of genes that probably are different.
Oh yeah, it was an extremely overwhelming process. The amount of data that you get with metatranscriptomes is insane. I think I had like around 10 terabytes of data or something like that. I didn't even know where to start. So then that's how I decided, well, maybe if I just focus on two big groups of course, like a ton of the data that you get are like housekeeping genes. For example, the genes that all organisms need to have, and if they don't have them, they die. And those genes, they usually don't go through drastic changes. They're usually always the same. So in a way, they're not that interesting in the context of climate change and how are fungi responding. So that was like the first thing that I thought. I was like, okay, all of these genes that are probably not under selection, I'm just going to discard. And then I was left with a ton of very interesting genes, but there's, I guess in a way, you need to be very strategic with this type of data because you rely almost 100% on databases. So you want to make sure that whatever you're looking at is in the databases. Otherwise, you won't be able to analyze your data. So then I was like, well, which set of genes are, have a robust database? And those were the carbohydrate degrading genes. The CASI database is actually amazing, so I use that database. And the other set of databases for metabolism is the PFAM. So I ended up using no. Sorry, I have a lot of data, and some of these data didn't make it to the paper. So for this paper, I ended up choosing the COG categories, but I do have data for PFAM too.
So the genes that code for CASIM, I guess they are also very interested in the case of climate change, because you want to look at carbon flux, carbon sequestration in the soil, and carbon degradation. You know? So that's and there is a lot of talking about this right now at the moment, about like the influence on microbes on carbon sequestration in the soil, for example, which has an influence, of course, on the level of carbon in the atmosphere.
Yeah, exactly. Yeah.
So using these genes, what was your conclusion about what is happening under this? It was a long-term experiment, right? It was 10 years.
It was it was 10 years.
Warming.
Yeah. So what I found was that the underwarming fungi were investing less in genes, or the transcription of genes for decomposition, and they were investing more in genes associated to cell maintenance. Basically just, whatever processes you need so that you don't die is what fungi were investing in. So they were really not investing in genes for decomposition of carbon, but rather investing in genes that will keep them alive.
I think it's because they're really under like this stress, and somehow, I mean, they just have to use this carbon for the metabolite, or they have to.
Yeah, I think so there's the conditions inside the greenhouses are extremely warm and extremely dry. And I think that those two factors combined are driving the high stress. So it's not the warming itself. So the temperature is a little bit more cozy for fungi inside the greenhouses, but it's also extremely, extremely, extremely dry. And without water, there's no decomposition. So I think that there's like the mix of those two factors that is making fungi decompose less. And what's really cool about this is that we carried out some decomposition studies inside the greenhouses, and we already knew that there was less decomposition inside the greenhouses compared to outside the greenhouses. But what was really cool about my new paper is that we were able to get to the mechanism behind that observation. So we knew that there was less decomposition, but I guess now we know why. And the reason is that we know that fungi are decomposing less because they're stressed.
Okay. But so in an environment which you have warmer temperature, but not necessarily drier, then that's not necessarily what you would not necessarily expect to have less decomposition rate. Or what do you think about this?
I think, well, based on what I've read in some other papers where they do have a warming experiment, but they also control for humidity. So they keep like water content in the soil, they keep it constant. They don't see decreases in decomposition. So I think that particularly in my experiment, like we're not able to tear apart exactly what portion of the results is due to warming itself and what portion is due to drying itself. But what's really, I guess, like something that's very important to keep in mind is that climate change scenarios come with drying. So the warming,
exactly.
like warming is going to cause drying.
In a lot of experiments, people talk a lot about temperature.
Yeah.
Very often, this is really linked with drier temperature. And a lot of times when you see effect of temperature, it might also be very related to soil moisture.
Yeah, exactly.
Okay, very cool. So can you tell us a little bit about what you're doing now? You're a postdoc now. So are you still working on these questions, or what are your new?
I'm working in related questions. So the one question or the one research area that I'm very interested in is fungal evolution and fungal adaptation in the context of ecosystems. So that's what I was working on during my PhD, and that's something that I'm still working now that I'm a postdoc. So what I'm looking at right now is how are fungi adapting to specifically deposition of nitrogen, and how that is affecting their ability to decompose. And so I'm still kind of like along the same lines. I'm still looking at how climate change is going to affect the carbon cycle. And I do that by looking at fungi and how they're responding.
Really cool.
Yeah.
Well, thank you very much, Adriana, for your time. Very nice to meet you. And we will be back soon with more interviews for the South American Mycorrhizal Research Network. Bye.
Bye.